everyone. Welcome, everybody. I'm always so excited when Dr. Sara Gianella presents to us because it's always something interesting and new and just does such a great job. I know a lot of people know her, but for those who don't, I just want to give a little background. Maybe she gave me, Dr. Gianella gave me a great summary. I'm going to try to make it a little shorter. So Dr. Gianella graduated from the University of Zurich after a residency in internal medicine and infectious diseases there. She began her fellowship in the Department of Infectious Disease and Hospital Epidemiology at the University Hospital of Zurich in 2007. She moved to UC San Diego in 2009 to work with Drs. Richman and Smith. And she joined the faculty in 2013, and she works between the UC San Diego Antiviral Research Center and her lab on the UC San Diego campus, where she does bench research, and she directs the San Diego CFAR Translational Viral Core since 2017. Her research is focused on delineating virus and host interactions to answer fundamental open questions in the fields of inflammation and organ damage and HIV persistence. Specifically, her current research efforts of her lab include understanding the interactions between HIV and co-infecting viruses, including CMV, and to investigate clinical complications related to persistent inflammation during effective antiretroviral therapy, characterizing the immunologic and viral milieu in anatomic compartments and tissues, for example, genital tract and CNS system, and then finally understanding sex-based differences in HIV pathogenesis and viral persistence, where she further expanded her research interest and focus to the transgender and gender non-binary community. She's received multiple awards, which I'm not going to name, and she's heavily involved in the AIDS clinical trials group on different committees and leading multiple trials. But above everything, Dr. Gianella enjoys spending time with her husband, Nadir, who a lot of us know, and her three wonderful kids, Emma, Noah, and Rebecca. So I will hand it over to Sara. As everyone knows, if you have comments, you can put it in the chat or message me directly if you'd like. I will give Dr. Gianella a 15-minute warning so that we can have some discussion at the end and only interrupt for clarifying questions. Take it away, Sara.
Okay, thank you so much, Jill, for the lovely introduction and for having me here. I would like to talk about barriers to cure HIV in women. I do realize as I presented this in 2017, a lot has happened in the last 4 years. And so I think that this warrants an update. So I have no conflict of interest. Here's an overview of my timeline as of my of the points that I will discuss. So importantly and of note, I will here mostly talk about cisgender women and sex difference as related to sex at birth, assigned at birth. And I think if you want, I can come back and then we can have a full total discussion about transgender and non-binary people. Okay, so there is a long history of exclusion of women in research in general. In the early 60s and 70s, women of reproductive age were excluded from research by default. I still remember when I was a kid, my mom talking about the scandal in Germany of thalidomide that was, right now it's used in the setting of oncology, but back in the years, it was sold as a sleeping pill. And women were using it even when they were pregnant, and a lot of kids were born missing some limbs, and that was a side effect of this medication. I think a little bit related to that bad experience, and a little bit because there was some patriarchal view of women. In the 70s, 80s, FDA explicitly excluded women with childbearing potential from early phase studies. And most of the research were conducted in males, and then results were extrapolated to women. And this, as you can imagine, is really not ideal because, of course, men and women are very different and as both biologically, and there is some difference that we need to account for. So in the early 1992, there was a committee was put together to determine ethical and legal issue relating to the inclusion of women in clinical studies. And then finally in 93, the NIH issued this Revitalization Act and finally said that women and minority must be included in clinical studies unless scientific rationale for exclusion. And since then, there has been increasing advocacy for more research for and with women. And this, of course, applies to HIV as well. So in HIV, women account for over 50% of people with HIV. This is particularly true in underserved countries like in sub-Saharan Africa. Over 60% of people with HIV are women. And young women in particular are disproportionately affected. And this is the leading cause of death in some of the African countries. On the other side, most cure-related research and clinical trial take place in countries where MSM represents the greatest proportion of the epidemic. And this is why a lot of clinical research efforts focus on cisgender MSM. And this is particularly true for cure effort. But that's the difference, the sex difference in HIV epidemiology is not only in international setting. Even within the US, the proportion of women affected with HIV changes across states. And you can see that particularly states in the South, they have definitively more.
Sarah, I think maybe if you're saying, I think maybe we're stuck on a slide.
Oh no.
Because someone messaged me and I thought we were going okay. Yes, now, oh, okay, now we saw the next one. Sorry about that, everyone. Okay, yeah, I think we're good.
I came to the office to make sure to use the landline so that we,
No, I think you're good now because I see the map.
Oh, okay. So this is the map. Sorry, and let me know if it happens again, please. Sometimes it happens, and there is a lot of people logged in, so maybe it's a Zoom issue too. And so you can see that the southern states are darker, which means they have more HIV infection among women. And then there is also a racial and ethnic problem within the US. And here you can see like there is an overrepresentation of Black and Afro-American women within the HIV population. Like the green bar shows the representation of overall women split out by race, and then the purple bar shows how many are diagnosed with HIV. And you can see there is a huge discrepancy for Black and Afro-American virus, and the other side, white women are less likely to be infected with HIV. So this was just to make the point that it's very important to study women in the setting of HIV, especially because minorities or people from underrepresented groups are particularly affected. And, not to state the obvious, but of course, cisgender women and men are different. And this is true at various levels. And not only in infectious disease, they are different related to autoimmune, cancer, vaccine response, and of course, infectious disease as well. So I wanted to show this nice figure from a paper in PLOS Pathogen. And here they summarized like a sex difference between male and female by viral pathogen load, incidence and prevalence of infectious disease and severity. And everything that is in blue, there's a male bias. This means it's worse for male. And everything that is red, there's a female bias. And you can see that there is so first of all, there is a lot of blue, there is some red. And if we look at HIV in particular, which is what we are interested in, it looks like there is a male bias when we talk about incidence, prevalence, and pathogen load, but then there is a female bias when we talk about severity. And so I wanted to show you some data to back this up. Here is a paper from 2001, but in the meantime, we had multiple papers showing the same thing, where we can see that during HIV infection, women have significantly lower HIV viral loads with comparable CD4 T cells. But when we look at disease progression in the same group of people, this was a large cohort, there is no difference. So this is very important. Like, as women have lower HIV RNA, but still progress to AIDS at the same rate as men. This is important because at that time, treatment guidelines were based on viral load and not CD4 T cells. And so you can see how women would have been disadvantaged. For example, in this cohort, at entry, only 37% of women compared to 74% of men would have qualified for antiretroviral therapy, but then both of them had similar progression to AIDS. And here, so this is a nice review from Monica Gandhi in 2002 that basically shows the same thing across multiple studies. So basically there is a difference in viral load between men and women, but then this difference does not change disease progression. So why do we see these differences? So I think there are for sure socioeconomical factors that play a role. Women, for example, have less symptoms during primary infection, so maybe they are less likely to be diagnosed and treated early. Women might experience more side effects from antiretroviral therapy, so they might be more likely to interrupt treatment. But putting all of the socioeconomical factors aside, which are very important, there is also a lot of biological difference that we need to consider. I took this figure from a review that Eileen Scully wrote in 2018, and you can see that she nicely broke down all of the possible causes underlying this difference. So for example, there are anatomic difference, the genital tract is different, the acquisition site is different, hormone modulates the risk differently between men and women. There are obvious genetic differences between sexes, and the immune cell phenotype is different, and we will talk a little bit more about it. There are also clear differences in latency maintenance, and we will talk about it as well. And then there are also that difference in microbiome that are currently under investigation, and how different microbiome composition at various anatomic sites might account for some of the differences. Let's talk right now about immune response. We know from other fields that women have more autoimmune disease and they have more robust response to vaccine. It looks like overall that the immune system in women is kind of more active than the immune system in men. In the setting of HIV, this is true as well. And in particular, multiple studies have shown that women have higher interferon response compared to men. And this is one figure from one paper, but this has been shown over and over. And multiple people have hypothesized that this increased interferon response play a significant role in this differential mechanism between men and women in the setting of HIV infection. Basically, this increased response early during infection causes a better control of the virus, but then over time, it is deleterious for the body, and this can also explain accelerated disease progression. So I'm not going into details about this in the next slide, but I did write in 2017 like a review, and I looked into different way how estrogen and progesterone can affect the immune system. And so immune cells have both estrogen receptors and progesterone receptor. And as you can imagine, the cycling of the sex hormone can affect all cells, immune cell subtype. Same for progesterone. But what about the reservoir? So as I showed you before, there is a lot going on. But there might be also some direct effect of sex hormone on HIV persistent. And if this is true, we would have to account for it because they are not stable, right? They change during menstrual cycle, during menopause, pregnancy, lactation. Women take hormonal contraceptive. So you can see how this complicates the picture. So I looked into all of the papers that have been published in the recent years about sex difference in HIV persistence. And this is like a summary of paper that looked at difference in HIV DNA. And you can see like results are not consistent. There are some paper that showed no difference, but the sample size were mostly small. And then there are a couple of papers that showed as a lower level of HIV DNA in female sex. I wrote a paper last year where we also saw a difference in HIV DNA between premenopausal and postmenopausal status. When looking at replication competent reservoir and HIV transcription, the picture is more interesting. So here, multiple people have shown that women, as compared to men, have lower level of replication competent provirus and also lower cellular HIV RNA expression. And this is consistent. So it looks like there is something going on with transcription in women compared to men. And here, I'm not showing you too many figures from all this paper, but this is one that makes the point from this Proger paper from last year, where you can see on the left, frequency of viral outgrowth. This means the virus who is actually replication competent, and on the right, frequency of total HIV DNA. And you can see there is a discordant results where females have higher level of HIV DNA, but lower frequency of replication competent HIV RNA. And this brings me to John Carnes' paper. This is kind of the seminal paper who has kind of led the research between estrogen and HIV transcription in the last year. So basically what they did in this paper, they did a screening of potential inhibitor of latency rehearsal. And they found that estrogen was one of the stronger inhibitor of latency rehearsal. This is in line with the data I showed you before, right? That women have less HIV transcription. And if you see, these are the data from the Carnes paper, you can see that these are all, so here you can see HIV RNA transcription in vitro with no stimulation. Here is if you stimulate all cells, they start transcribing HIV RNA. This is if you stimulate all cell and add estrogen on the top of it. You can see that HIV transcription is significantly lower. And similarly, if you stimulate cells and add an estrogen receptor antagonist, the HIV transcription increases. SAHA, it's one of the kick and kill drugs. So you can see if you use SAHA alone, cells start transcribing HIV, but then if you do SAHA and you add an estrogen receptor antagonist, HIV transcription is higher. And this estrogen effect is sex specific. So here they took cells from cisgender women and cisgender men, and they did a similar experiment. And you can see here how profound the effect of estrogen is on HIV transcription in female, but not so much in men. So let me give you a summary of what I showed you so far. And it's that the important difference exists between cisgender men and women in the setting of HIV persistent, and that estradiol at peak menstrual cycle level is a potent inhibitor of viral replication, which possibly interferes with kick and kill strategies. So my friend and colleague Eileen Scully, based on this data, designed a clinical trial, ACTG 5366, where she was basically taking women, giving them a kick and kill drug, vorinostat, and then half of the women were randomized to receive tamoxifen, which is an estrogen inhibitor, yes, sorry. And the clinical trial finished, I think last year or a couple of years ago. The results are still unpublished, but she did present some of them during her plenary at CROI last year. And basically, tamoxifen did not enhance vorinostat effect on HIV transcription, which was surprising. But I have to say that only postmenopausal women were allowed to be enrolled in this trial. This because the FDA and ACTG raised safety concern in giving these drugs to women in reproductive potential. And this might be one of the causes why we didn't see an effect. So maybe we were looking at the wrong population. And, there has been a lot of controversial about this, because there were women that really wanted to enroll in this study and that they were not allowed to enroll because again, we were worried about their safety, which we can open a full discussion about it, but I think people should be able to make their own decision about how much risk they want to take on, as long as there is a proper informed consent.
Sara,
yeah.
Tamoxifen is a selective estrogen receptor modulator. It's not a pure antagonist.
Okay, yes, thank you so much. I wasn't sure anymore about that, but thank you for clarifying it. Okay, so, but one thing that we learned from this study, and this is the first time the ACTG has ever done a women-only study, is that, and there was a lot of concern from the leadership that Eileen was unable to enroll it, and they even made her put in some stopping rule, like if you cannot enroll women, then we will abort your trial. But in reality, it has not been a problem. Like, they enrolled 31 women participants quickly and without any difficulty across the United States. And most of them were Black. They had a median age of 57, and they have been on ART for a median of 7.5 years. So the same group published a nice perspective led by Karina Dubé, where they did some qualitative interview for these women to see how they thought, also how they felt about being enrolled in this trial. And overall, all women had a very positive experience participating in this trial and will recommend the study to others. I think this is a huge, despite the fact that the trial was negative, this is a huge win and a proof of concept for the ACTG that we can enroll women in clinical trial if we work hard enough and do the outreach. Okay, now I wanted to show you some additional data about the effect of estrogen on HIV persistence, and these are data from a CROI abstract that I will present next week at CROI. And basically what we did here is to further study the effect of estrogen on HIV persistence. We took samples from women that went through reproductive aging. Basically, we started at 45, and then we followed them for about 10 years, assuming that they will have a decline in estrogen. Of course, this is a retrospective study. So we selected sample from 2 cohorts. One is from ALERT, that's an ACTG longitudinal cohort, and one is from the WISE, which is another longitudinal cohort. So we had 409 ALERT sample from 59 women and 31 men, and then we had 49 sample from 15 women from WISE. The reason why we went to WISE is that they have a better characterization of gynecologic history. And so for the ALERT, we had to rely on sex hormone to classify our people, while for the WISE, we had very precise data and we were able to select our women across reproductive stages. So, so these are the estrogen and FSH data from the ACTG cohort. So as expected, we have overall a decline in estrogen and increase in FSH, while the men remain stable. This means most of the women somehow were going through reproductive aging during this time window. And we did measure HIV DNA and cell-associated RNA in all of this sample. And we observed a significant decline of total HIV DNA over time in both men and women, but you can see that the rate of change was different between sexes. And specifically, women had significantly slower rate of decline as compared to men. And this was becoming more pronounced with age. And so basically women have almost a plateau as they age compared to men who keep going down. But then when we look at the level of replication competent HIV RNA, we can see that it increases across reproductive stage significantly. This is a huge surprise because technically we are used to see, as when we look at the general population of people with HIV, the HIV reservoir is declining. But when we look at women in reproductive aging, it increases. This is kind of this was a surprise to us. And I think it's important to know, and it's important to understand why this happened. So basically, the conclusion of my study is that there are sex-specific HIV reservoir dynamics as people enter reproductive aging. And while total HIV DNA declines in both sexes, there is an increase in replication competent virus in women after menopause. And this warrants more study. John Carn and myself and Eileen Scully and others have submitted an R1 to study this further and to understand the mechanism underlying this surprising increase of replication competent reservoir over time in women. Okay, so after showing you all of this data, so we all understand that one cure might not fit all, and that eradication strategy needs to account for estrogen and perhaps all other hormones that we don't know yet about. And estrogen antagonists are well established, like for example, tamoxifen, as I showed you before, and I wouldn't give up on it only because one small and clinical trial showed no effect. I do think that there are, as we need to keep exploring this venue. So I wanted to now show you, talk a little bit about, why women have not been historically enrolled in research, and then finish up trying to give you some thoughts about how we can do better. And here I wanted to show you a couple of papers that have actually looked at how we are doing in enrolling women in research. This is a little bit of an older review from Rowena Johnson, where they looked at 125 studies with almost 13,000 study participants, and 11% were women. 32 study reported no women enrolled. And you can see that some study like cell therapy or reactivation, like basic core cure study, often exclude women. Similar data have been shown in a more recent review from Liz Barr, where she also interviewed multiple sites. This was an international study. She saw, like, in, I think here, these are data from almost 3,000 participants. And she found 17%, but this is in international setting and across all cure studies. But we still can do better to enroll women in study. I do think that we need to do better. We need to do better not only in the, like in large clinical trial, but at every stage of HIV research. So, for example, early phase clinical trial, those are tricky, right? Because there is a high or unknown risk, but no clinical benefit. And so many people argue that we should not include women of reproductive potential, or we need to make it very hard for women of reproductive potential to enroll in this study, because we put crazy contraception requirement, and sometimes this requirement that we put prevent women from joining study, even if they are interested or willing. In the setting of preclinical research, for example, animal research, even here we have bias towards more male animals, because women are often preserved as a breeding stock, and also female animals are less as harder to standardize because of hormonal cycling and all, also in particular hormonal cycling. So even at that level, basically this bias might obscure key sex difference that can guide clinical studies. And similar, even in vitro studies, we need to make sure that when we collect donors, we always balance cells from female and male participants. Because I cannot stress this enough, a failure to enroll women in every step of research will create huge knowledge gaps and a missed opportunity to learn about sex difference. But what are our challenges? I'm not saying it's easy to enroll women. Like, women have competing priority. A lot of women are the primary care provider for kids, and they work multiple jobs, and it's very hard to get them to the sites to perform all of this research on them. Collect sex-specific sample is a challenge. Like, if we need to collect genital secretion from a man, that's easy. If we have to do a pelvic exam in a woman, that's not as easy. Need to sample through the menstrual cycle, like we need to time the visit to the menstrual cycle, and we need exactly to know at what stage of the menstrual cycle women are. And so we need to establish new recruitment practice. Like, it is more complicated to enroll women. I'm not saying it's not. I'm just trying to explain that it's important to do it. This is another paper from Karine Dubé, where she reached out to women and men and asked them for their willingness to participate in HIV cure-related study. And you can see that overall, female were less likely to participate. Like, the yes is in blue, the no is in red, and the I don't know is in gray. Like, there are some difference between male and female, but still over half of women, or half of women, were willing to enroll in HIV cure studies. So there is some willingness and interest from women to enroll in these studies. So Liz Barr, some time ago, also put together a consideration for cure research with women, and she queried women who are part of ACTG sites and asked them what's the reason why is it hard for you to participate in HIV cure research? And again, competing priority, stigma, lack of education about clinical trial, unwillingness to serve as a guinea pig were brought up, but competing priority was number one. Okay, so my conclusion is that sex-based difference affect the natural history and immune pathogenesis of HIV. So we know it, and we also know that it also affects the HIV reservoir, in particular HIV transcription. So we need to do targeted enrollment of women in clinical trials, and we also need to do careful sex-based analysis. We need to design sex-specific approach to HIV eradication, and we really need to look into it. And we have to consider cis and transgender women and the role of exogenous and endogenous sex hormones. So here are the recommendations that we came up with. First of all, sex-based analyses are imperative at any stage of clinical research in order to understand biological difference in HIV reservoir dynamics. I'm actually glad to see that more and more journal and conference require sex-based analysis, like at CROI. This is a requirement, and if people don't do it, that can be a reason for your abstract to be rejected, or they come back to you and ask you to present the data. We, as part of the ACTG women's group, we did do an analysis of CROI abstracts to see how many adhered to this rule, and we were still able to find a lot. Now I can't remember from the top of my head, but there was a big percentage of abstracts and oral abstracts that were not following the guidelines, and we actually wrote a small report to J8 last year and submitted a letter to CROI to try to have them enforce these guidelines. Clinical trial protocol can be written to include sex-based comparison. So in all of my clinical trial, I make it, I am committed to perform sex-based comparison, and I always put in a minimum number of women that need to be enrolled. In both 5383 and 5355, we have a minimum of 30% women, which include both cis or transgender women that need to be enrolled. And if necessary, we will stop enrolling men and we will keep enrolling women if that's what we have to do. But I think it's important because we need to be able to make this, to see the differences. Targeted enrollment of women is necessary at all phases of clinical research. This is what I told you before, like not only in clinical trial, but in early stage clinical trial, animal study, basic research. We always need to think about sex. And when I review paper for journals, I always point it out. And there are, especially basic research paper, they mostly do not tell you if the cells that they use for their experiment come from male or women. So publication must always report the number of women enrolled, even if when it's 0. I think that's important too. And I have published a paper with 0 women, especially here in San Diego, where most of our, a lot of our patients are men. It's not easy to enroll women. So I don't think it's necessarily wrong, but we have to say it and be transparent about it. And then we need to engage the community and other women to make sure to target the right population. Of course, so I concentrated on cisgender women. The same consideration apply to transgender women and to gender non-binary individuals. Exclusion of women based on gender identity is a problem. And so the next step will be to make sure that we enroll enough transgender women, because they might be affected differently, because exogenous sex hormone might have an even different effect on HIV persistence. We are right now designing another ACTG trial where I am serving as the virologist, which is a transgender women only ACTG trial, where we will be looking as our primary outcome and the interaction between antiretroviral therapy and sex hormone. but we will have a secondary objective to look at the effect of sex hormone on HIV persistence. Okay, I'm done. I wanted to acknowledge, of course, Eileen Scully and John Carn and Karine Dubé and Katie Godfrey, so they are all my friends and passionate like me about women's health, Liz Barr and Lisa Dawson, and many other people that have helped me come up with all of these slides and material. Okay, let me see. I see there is already a couple of comments in the Jeff Taylor. It all boils down to the study team and the sponsor committed to enrolling statistically significant number of women. Yes, I totally agree. Like, I do think that the study team and sponsor need to commit to enroll more women. Like, and as I said before, if we need to stop enrollment for men, that's what we have to do. If we have to keep study open longer, that's what we have to do. I understand it's not always cost effective, but I do think it's important. And then Miley said that we need to implement recruitment and research strategy that make it easier for women to participate, such as childcare, flexibility in scheduling appointment, and trauma-informed research. Yes, I totally agree. Those are very good points. A lot of people, so childcare will be crucial, but also trauma-informed research and flexibility in scheduling appointment that will help. And I know, for example, the HNRC, I'm not advocating for opening the AVRC on Saturday, but I know the HNRC is seeing people on Saturday, and I think that helps.
Yeah, I think Miley's point is so important. It seems like a lot of studies, collect the same data, or we find the same thing in that women say we have competing priorities. And then we don't do anything to sort of address those competing priorities. We know that, we know they have it. So, obviously having things like childcare and flexibility in scheduling appointments, all are great. I wonder if we can go a step further. Can we take research to people? Can we go there? Can we go where they are? Obviously that won't work for everything, but I just feel like we come up with the same conclusion and then we don't make significant changes. And I see, anyway, I was just thinking that as you talked.
Yeah, so Jeff and Susan, both of them point out that if we want to recruit women in our study, the budget needs to reflect this effort. It all boils down to money. So, for example, in our transgender only ACTG study, which is right now relatively early, we are already thinking to ask extra money for extra outreach within the ACTG, but that this has historically not been easy because people, so that's not where the money goes. And I think this is where the money needs to go if we want to improve our reach to particular groups that historically have been underrepresented. Like, our recruitment machine works very well for men, for white men in particular, but we need to be willing to put in the extra effort to reach out to other populations, and we need the money to do that. So I totally agree. And I'm curious to see how our transgender only study will go, and if we will make it. Like, Miley is particularly disappointed with the clinical trial around new HIV meds. I hear you, Miley. So if people from industry,
they did a very nice job for long-acting injectables. They did a very good job enrolling cisgender women for the FLAIR and ATLAS studies for long-acting cabotegravir and rilpivirine. It was very intentional, but it had to be very intentional.
Yeah, but I think it's important. It's very important that we do it consistently.
I think one thought is to partner up with the clinicians, whether it be OBGYNs or clinicians seeing women, because I think the traditional, all the points that have been made are excellent. And I just think that the traditional model of women coming to an extra visit for research when they've got kids and 2 jobs and a husband and a car that's broken down is unreasonable. And I was just even thinking, Sara, about the comment of wanting to know exactly where women are in their menstrual cycles. I mean, that's super complicated just from how they vary from month to month, but women don't know. And I wonder if that's even, if you know that information, how helpful is that in regular clinical practice when women don't even know where their cycles are? So anyway, I think there's even broader, life is messy for women and not as neat and tidy. And I think maybe the research needs to be a little bit messier too, and account for that.
I totally agree. And, if that's the case, then we need larger sample size, which basically, because, if you have messy research and you have multiple confounders, then you just need even more women to make the point. I also see that somebody is proposing that maybe we need to do a study to optimize recruitment methods for women, which I think it's a very good point. And the ACTG has just put out a call to do a social behavioral committee as part of the ACTG. And one of the mission of this committee will be, to create a survey for study participants to optimize recruitment methods. So I think that the ACTG is doing some step in the right direction.
But again, you'll still be missing all the women who don't enroll. And so that survey might be better done in a clinical setting, because again, you're only going to be surveying the women who make it.
Yeah, that's totally true.
Well, I was going to say something about that because I've enrolled several hundred women now on different studies, and I think the problem with getting it to the clinical sites is I've tried to do that, but it doesn't integrate well and it doesn't enroll well when we've tried to work with clinicians in different primary care sort of settings. And so, in the end, it was really more that what was successful was, again, just more intensive recruitment and a lot of support and providing enough, financial support too for them to come to their visits, and a lot of follow-up. And that kind of worked, through several of my women's studies.
Thank you, Sheldon. Another thing that I think Jill brought up before is that maybe we can bring the research to the people. This is what we do with the last gift, right? Because as part of the last gift, our people are terminally sick and we don't want them to have to come in the AVRC. We basically go to their house, and then Susanna and Stephanie, they do their blood draw, they ask them hours of questionnaires. It can be done. It just requires more people, right? Because then visits are more intense to be organized. But I will totally enroll in a trial if somebody comes to my house and draws my blood. But I will probably not enroll in a trial if I have to go somewhere right now. I actually enrolled in a trial and I missed most of the visits because I just cannot get it done. Sorry.
But this is Connie. I think the point you're making is that with taking the trials to their homes is something we're starting to learn in the context of COVID research as well. Since a lot of people who've been in quarantine and can't come into a research site, we've contracted with CROs to actually send nurses out to the house to draw blood, to do a clinical assessment of the patients, to gather data. And there's no reason why we can't come up with schema for clinical trials that aren't COVID related that have to do with that, if we're specifically put that into budgets and into the monitoring parameters of the trial. I've actually been in contact with a couple of companies who manage home care for the elderly who would like to get involved in doing sort of home visits for clinical trials, and have approached me about how they can get into that as a strategy for their business model. And I think that's something we should be considering for the future.
I love it, Connie. Yeah, I think that's the way to go. And I'm thinking that we should actually, especially for the transgender only ACTG trial, we should just budget that in and see if we get pushback.
Well, I think the big point that's been made is this is obviously more expensive to do if this is, if that's what you consider. So that has to be factored in. I think the other point is, I think for a lot of people in research, but maybe particularly cisgender women, is that one size doesn't not fit all. There are going to be some women who do not want you coming in their home for various reasons. And so I think a lot of this has to do with being flexible as much as possible. And I think that was the point that Miley was making. How do we work for you as a study participant? How do we make this work best for you? And potentially have different ways to do it. Obviously that's complicated, but to me, just like with anything, giving people more options probably helps with what you're trying to do, particularly in this setting.
Agree. So somebody also said that maybe we can also give people telemedicine equipment and training, and do some of the visits remotely, but then we have to make sure that people have high-speed internet and smartphones, which of course a lot of the most underserved participants might not even have.
We've actually done that with the COVID trials too. And basically before they were discharged, these were the inpatient trials, but before they were discharged from the hospital, we gave them a iPad or a smartphone that connected back to our research site. And we were in contact with the patients at home through telemedicine using their smartphones. That has been an avenue of research more recently applied to COVID, but could easily be applied in this settings as well. You can't obviously do blood work that way, but you can certainly collect clinical information and follow-up data that way.
Yeah.
We actually budgeted the smartphones into the clinical trials budgets.
That's the way to do it. I agree.
The silver lining of COVID, all of the lessons we're learning and how we can apply them to other research.
Any more questions or comments?
Sara, this was phenomenal. So thought-provoking and obviously a great dialogue. And you presented it in such a clear and concise way, as always.
Thank you.
So thank you so much.
Okay. Have a nice evening, day, and weekend.
Oh, evening. Wow. We're jumping. Thanks, everybody. Try to fill out the evaluation form if you can, and we'll see you next Friday.